I will talk a little bit about practical approaches which we are doing in my lab. And I need to declare a conflict of interest. I'm a scientific advisor of tissue gnostics. So I will first go with you a little bit what can be done using the tissue fax. I have the tissue fax confocal, which in principle also permits three-dimensional imaging. And what I will show you overall is here some data, so how we investigate Alzheimer's disease mechanism in mouse model. So for this, we use the APP-PS1 mouse model. And this APP-PS1 model is basically has two mutations, and it's a model for early onset Alzheimer's disease. Yes? And what you can see here, that you have plaque deposition, and you have also the features of neuroinflammation. Yes, and the idea of this model is that you can study alterations in behavior, and you can also study all the logical alterations that you see, such as inflammation, plaque formation, neurodegeneration, and cell death. So here we use the tissue fax for imaging beta amyloid deposits in the hippocampus of this animal after 6 months. And what we can see here, that we did a quantitative imaging where we can compare in this APP-PS1 Alzheimer model, so the number of plaques, and do a quantification of that. So all the data what I show here, they have been published in peer-reviewed journals. And now also, what the aim of this work was, what we published in Stem Cell Reviews and Reports, was to check what happens if we transplant these animals in the hippocampus with neural stem cells or with mesenchymal stem cells. And what we could see here, that we have actually, in these cases, we have a reduction of this amyloid plaques in the hippocampus. So it shows that we have an improvement regarding this, so that we get down with amyloid plaques, and also probably what we are getting down with inflammatory parameters. Okay, you see this here with neural stem cells here in A, and we see the same thing here. This is the second experiment. On the left side, there was the number of plaques. On the right side, we have the areas of plaques, so we have a clear reduction of this. So now, what happens is during Alzheimer's disease, microglial cells get activated, and they will basically secrete cytokines and neuroinflammatory parameters. But then these microglial cells will undergo aging and also senescence. And this I will show you in the next slide, fragmentation of microglial cells here, for example, in the cortex. And we imaged this with tissue fax. You can see here we have an I/1 staining in this APP-PS1 mouse, where you see here stained the activated microglial cells as an original image. And now we will identify cell bodies with filaments, cell bodies in yellow, with filaments in green. And this is now followed by an analysis of this fragmentation, what we can see. So we have distant filaments, we have long attached filaments, and we have also short attached filaments. So if we have the distance filaments, so it means that they have been fragmented, so these microglial cells have undergone fragmentation. This we can see here in different colors, but we can do a quantitative analysis of this, where we see the percentage of distant filaments, the percentage of long, and also of short filaments. So it means tissue fax is not only able to say you how many microglial cells you have in this slide and how many of them are activated into a pro-inflammatory M1 stage, but you can also say how long are the filaments, and you can do a quantitative analysis of these filaments and say there's a difference to the control non-Alzheimer mouse. It's basically, you can do a backward connection, for example, here on the long attached filaments, which you see here in blue. So it means not only you see a separated dot plot from your image, but you can go from your gated area if these are really the cells which you define with long filament. So it means that would be microglial cells which have not undergone senescence and fragmentation. So one point was fragmentation, and the second point would be how many activated microglial cells you would have. In this case here, we did the same thing as the microglial cells, which we detected the nucleus by using the DAPI marker, and then in the second field, we detected IBA1, and basically as a marker of activation of microglial cells. And now we can go here for a gating analysis and determine how many of these cells are activated, are either positive, and then again we can look at different types of microglial cells, such as, for example, void and ramified microglial cells, and also round microglial cells, which would be in another stage of activation. And like this, we can see it's a quantitative plot. We can see how many of these cells are positive. In this case, we use a mouse model, and we injected this mouse model with beta amyloid. This is another possibility of creating an Alzheimer disease model. And then we could see, in this case, after amyloid beta injection, when we can measure here the percentage of cells which are positive for IBA1, and the number and the percentage and the number of round, so it would be an activation status of IBA1 positive cells. So besides of that we are measuring the stages of microglial cells, are they activated or not? Are they undergoing fragmentation? And we can also measure directly neuronal projections, which is important in developmental biology and brain development, but also with neuronal projections, very extremely important if we look at neurodegenerative diseases and also in neuroregeneration. And as we can see in the next slide. So this shows here a migrating neurosphere. So it means these are neural stem cells collected from a mouse or rat embryo and put into culture and induced to migration. And what you can see here, that we have in green and red, we can see that we have beta-3 tubulin staining and GFAP glial staining. So we have neuronal and glial staining. And we see here something like what would be a mini brain, because here happen the same processes which you normally see during brain development. And now we go to analyze this by tissue cytometry. So we have an original image. So we will trace the neural outgrowth as the filament structures after staining for beta-3 tubulin, as I showed you in the last slide. And now we have an original image, and we get the filament structures. We can quantify the number and the length of these neurites. And then with neurites that we have identified, we can do backgating and what we have defined in a gate, and we see what is inside of this gate. So it's beside the staining. It shows you also the size of these neurites. So now I come to a real research project that we did in my lab. So what my lab has found, that neuronal differentiation is promoted by bradykinin. Bradykinin is a vasoactive peptide, which before nobody had shown, but this may affect differentiation of neural stem cells. But the idea was here, again, using the same model that I've shown you before. This is an excellent model to be analyzed by using the tissue-fax system. So you dissect the embryonic brain of a rat, of a mouse, and then you get put individual cells of the telencephalon into culture, and each of the cells will form here a neurosphere by clonal expansion. So after initial proliferation, you induce the cells to differentiation by removing the serum, and then the cells will differentiate into neurons, astrocytes, and oligodendrocytes. And this is an extremely nice model because it shows you all what you will also be interested in vivo, such as proliferation, migration, differentiation, maturation, and also synapse formation that you can analyze by using tissue cytometry. And looking like that, we can see a cell migration. So this would be a DAPI or staining, for example. And then you can see neurogenesis, you can see glial cells, and then you have this image that I had shown you before, and you can analyze all of this by using tissue cytometry. And the idea was that we used this bradykinin receptor, this is a G protein coupled receptor, which would classically reduce blood pressure. So we go much beyond of this, and this work was a long time ago already recognized by FAPESP, and they published here something in their journal and saying, okay, bradykinin makes neurons. And yes, and it is so, and it makes also neurons in culture, and it makes also neurons in vivo. And now, using the tissue fax system, we can again detect migration, right? Migration of neurospheres out, and how is this migration radius affected by bradykinin or by its inhibitor HOI140. We see that bradykinin induces neurogenesis here in red, and if we inhibit bradykinin receptor, then we see here in green that there is a lot of GFAP, a lot of astrocytes, but there are much less neurons. On the other side, bradykinin stimulated cells have less astrocytes. And this can be done looking at this affects not only pharmacological models, but also affects transgenic model if we have a knockout mouse without the bradykinin receptor. Okay? And the idea was that bradykinin would induce differentiation to neurons. And through an enzyme here that activated that is ERK. So it's a MAP kinase which induces gene expression. So now we come back to tissue cytometry. We sent this paper by manual analysis to Journal of Cell Science. And the problem is we wanted to see something in vivo. They say, okay, that's fine, what you say here. But could you show that bradykinin also stimulates neurogenesis in vivo? And for this, we injected bradykinin into the hippocampus of postnatal mice. And then we did tissue sectioning, and we used here bradykinin, the inhibitor of the bradykinin receptor HOI140, and U0126, which is an inhibitor of ERK. And now we are looking for tissue fax analysis, counting neural stem cells, which are very rare in the hippocampus. And what we can see here, we got here with dot plots. Now we have a couple of cells here which are positive, but we can say that bradykinin forces these cells into the cell cycle as positive for Ka67 staining, while HOI140 and the ERK inhibitor inhibitors. So using this tissue fax technology with segmentation analysis, we could show that bradykinin does this also in vivo. And I see here, so that we have a very small percentage. We see Ki 67 cells, which I show you example. It is a very rare event, but we were able to quantify this using this technology. Okay, so now I switch to our field. The same thing, I'm looking into bradykinin in developmental events. So now we say that bradykinin promotes differentiation of muscle cells, skeletal muscle cells. And if we look at this slide here, it may be because this is heavy chain. And what we would do now is that we would segment the cells here. So if muscles are going to differentiate, what happens is that they form syncytial cells which have more than one nucleus. So what we see here in the beginning, so there are single nucleated cells, and when there are multinucleated cells, and you see that we are able to detect them by using the StrataQuest program of tissue fax. And in the next slide, what you can see, that we have a very small difference Here, if we look at the number of nuclei, so where we have a difference here, if we inhibit bradykinin receptor, what you see here, number of nuclei per square millimeter. So here we have a difference, yes? And we see also that here, treated with HOI, that we have more cells which have only a single nucleus. If this happens in the presence of HOI 140, then we can say the cells, they stay at the single nucleated stage and they are not going to differentiate. So we can do a lot of analysis altogether. And we can also measure the size of the cells that we have and all type of this stuff. And further on, what we can do also using this technology in applications in cancer research. For example, with a collaborator in Rio Grande do Sul at the PUC University, we investigated what is about papuinergic P2Y2 receptor in esophageal cancer. The papuinergic receptor is a receptor activated by extracellular ATP. And what we could find here, especially for this protein, we used here tissue section. We're not anymore talking about cell cultures. Now this is a tissue section where we make a nuclear detection mask, and then we detect the P2Y2 receptor. And this we do in esophageal biopsies of patients' immune stains for P2Y2 receptor. And basically what we could see, if we compare carcinoma, squamous cell carcinomas, compared to this tumor, esophageal adenocarcinoma, we would have a reduction if we look at esophageal squamous cell carcinoma, which has less P2Y2 receptor than the adenocarcinoma. And here we can measure the area, the mean of the positive cells, and we can also measure here the mean intensity of the staining of single cells for the P2Y2 receptor. And finally, we have published a large number of papers, and some of them also in high impact factor journals like Molecular Psychiatry. And for this, we were last year awarded from Tissue Gnostics as an excellence center in tissue cytometry. So this owes this is owed to my scientific production in publishing high quality and high impact factor papers, as well as of my colleagues Claudiana Lameo and Nicolas Hoch, who are working with the same technology in my institute. And finally, I would like to thank my group. So my postdoc, Talita Glaser, she's a specialist in tissue cytometry, but all of our postdocs, like Agatha Oliveira and also Roberta Andrejev, we are also working on tissue cytometry in different types of models. I showed you the Alzheimer disease model, but I could also show you the same thing on Parkinson's disease, that we work in my lab, and also on Huntington disease. And Michelle Pilar, she is in collaboration with me with technology to develop her work at the Federal University of Santa Maria. And she did in my lab the work on bradykinin in neuroregeneration, and also we are working on the topic of Parkinson's disease. I would like to acknowledge Professor Beatriz Longo of the Federal University of Sao Paulo. I work with her on the Alzheimer model, so she provides the animal models, and also Fernanda Moroni. I showed this data in collaboration with her on esophageal cancer. And finally, I would like to acknowledge the funding agencies, and I'm thanking Tissue Gnostics for the collaboration and for support. So thank you very much.